Know that mitosis and cell division is a complex process, and the resulting daughter cells must be genetically identical to one another in order for the process to have occurred correctly. So that means that the cell cycle is going to have to be tightly regulated by certain molecules to make sure that everything is occurring properly. So in this lesson, you're going to learn how is the cell cycle regulated to ensure the creation of genetically identical daughter cells. When a cell goes through the cell cycle, there's three options of what the cell can do. Excuse me, what the cell can do once it hits certain points in the cell cycle. The first fate of a cell, meaning like what its outcome is that if after cytokinesis, when the two daughter cells are created, those daughter cells can go back into the cell cycle. So if those cells need to keep dividing, then they're going to return to the cell cycle at interphase and go through the process of G1. Then when they get the signal to divide, SG2, mitosis, cytokinesis. So of course your stem cells are going to be examples of cells that actually continue back into the cell cycle, especially those cells that stay in the niche or the niche where they have to reside so that they can keep dividing and keep replacing lost or damaged cells that are in your body. Now, the second option of a cell is that it can enter a phase called G0. So during G1 phase of interphase, if a cell is no longer going to divide, the cell is going to leave the cell cycle, and you can see this happening at the bottom right-hand side of the screen where the G0 is, and it says cell cycle arrest. The cell is going to leave the cell cycle, and it's going to go to a non-dividing state, never to return to the cell cycle again. So examples of cells that go into G0 phase are those specialized cells that we talk about that stem cells are going to create. So when a cell leaves the cell cycle and goes to G0 phase, it's usually because signals were received that that cell needs to actually specialize and turn into something like the muscle cell that I showed you in class. That's so highly complex that it will never be able to divide again. It's just too far gone. It's too crazy. It can't divide. Other examples of cells that go into G0 phase would be things like your nerve cells, the cells that send signals to your brain. Also, your cells of your different tissues, so like your heart cells or your stomach cells or the cells of your intestine, all of them will leave the cell cycle and go to G0 phase, never to divide again. So G0 is called cell cycle arrest because the cell leaves the cell cycle and it stays in that non-dividing state for the remainder of its life. The third and final fate of a cell is called apoptosis. Apoptosis is programmed cell death. That's why I have a picture here of a cell sitting on a chair with some dynamite underneath of it. Sometimes there's going to be issues that happen with the cell cycle where it doesn't replicate the DNA properly, or it doesn't separate the DNA properly, or the cell doesn't split properly, and there's going to be an issue with the cell, and the body can recognize that issue and cause the cell to die. If there's issues in the cell, we don't want that cell to keep replicating because then it's going to pass on those mutations to all of its daughter cells, and you'll have what can turn into a tumor or turn into cancer. So our body has this nice mechanism called apoptosis, so that when a problem in the cell cycle arises that cannot be repaired, the cell is going to die off and it will no longer replicate. Another example of apoptosis is, many of you may not have known this, but many of you may. You are actually webbed. Your feet and your hands are webbed when you are in early development of the fetal state. And what happens then is that the cells that are part of the skin that's between your fingers and toes actually undergo apoptosis, and those cells die off so that you no longer have webbing. But there's some people in this world that actually do have a little bit of webbing in between their fingers and their toes. That's because they had a problem with the apoptosis of those skin cells between their fingers and toes. The apoptosis didn't happen, therefore they have the webbing still in between their fingers and toes. So how is it that the cell is going to make sure that the cell is dividing properly? How can it decide if a cell needs to go through apoptosis? How can it decide if a cell is going to go into G0 phase, never to divide again? Well, the cell cycle checkpoints that are part of the overall cell cycle are going to check the cell cycle at certain junctions in the process to make sure that everything is going the way that it should. The proteins that are responsible for regulating the cycle are called cyclins, which is very cute because proteins that regulate the cell cycle are called cyclins, cyclins, very cute. So what happens is that if the cell is ready to move on to the next checkpoint, and notice down in the bottom right-hand screen, there's a picture of the cell cycle with little stoplights. Those stoplights represent the checkpoints that occur during the cell cycle. So when the cell is ready to move past those checkpoints, then the signal called cyclin actually increases in concentration in the cell, and it actually helps push the cell past that checkpoint and gets it to the next part of the cell cycle. So as you can see there, there's a checkpoint after G1. Well, not really after G1, but this checkpoint has to be passed for the cell to move on to S phase. So of course, things like DNA is going to be checked there and make sure that the DNA is okay so that bad DNA doesn't get replicated. Another thing that happens at G1 checkpoint is that the cell makes sure that the cell is large enough. We've said that G1 is the growth phase, so if the cell is not large enough, we don't want it to divide. All right, there's another checkpoint then partway through G2, and that checkpoint must be passed for the cell to go into mitosis, which is, of course is nuclear division. At this checkpoint, we might be checking the DNA again because it's just been replicated and making sure that the cell is ready to divide. The last checkpoint is going to be during the M phase or mitosis checkpoint. This checkpoint is going to make sure that the spindle fibers have attached to the chromosomes properly, because if you think about it, if the spindle fibers are not attached to the chromosomes and when they start to pull them apart in anaphase, those chromosomes are not going to separate in the way they should. So then you end up with daughter cells that have too much chromosomes or too little chromosomes, and problems arise in the cell cycle. What you guys are responsible for doing today is the cell cycle regulation map activity. This can be found on the second page of your cell cycle regulation notes. It just looks like a big picture of the cell cycle with some stop signs on it that represent the different cell cycle checkpoints. The first thing you're going to do is you're going to place the green cards that you'll get in an envelope on the map, and those green cards represent interphase and its different phases. So make sure you put them on the correct spot on the cell cycle. If you need to, please take out your mitosis notes and go back and figure out where those cards need to go. The second thing you're going to do is put out the pink cards. The pink cards are cell division and its phases. So that would be mitosis and cytokinesis. Last but not least, the yellow cards. This is new information. The yellow cards represent the cell cycle checkpoints. You guys need to read what the cell cycle checkpoints say they do and then figure out where they belong on the map. What stop sign do they belong at? Don't just guess on this. Really think about what the descriptions are telling you. Really think, does it make sense if spindle fibers being checked would attaching to chromosomes, does it make sense that that would be happening in G1? No, of course not, because mitosis hasn't begun yet. So make sure you really think about it. When you think you have all your sets of cards correct, you're going to raise your hand as a group, and the substitute will come and check your answers. The substitute teacher, however, is not going to tell you what to fix. You need to make sure that you think about it and think it through and put the cards down correctly on the map the first time. The substitute may say you're wrong and you have to fix it, but they're not going to tell you exactly what to do. All right, when the cell cycle map is complete and it's completely correct, you're going to want to fill in all the information that's on the cards from your cell cycle map onto your cell cycle regulation diagram in your notes. So make sure you put all the information down. Don't just write spindle assembly checkpoint or whatever. Make sure you write down what the spindle assembly checkpoint actually does. After you're finished with your map, then you're going to hypothesize what would happen if certain checkpoints fail to work the way that they should. You're going to predict, for example, the first checkpoint when it says the cell, or it checks to make sure that the cell has grown enough. If a failure happens there and the cell hasn't grown enough, then you end up getting two small cells as the daughter cells without the proper number of organelles and nutrients and all that kind of stuff. So those daughter cells are not going to make it. They're probably going to die by apoptosis. So you're going to make two predictions for each checkpoint. You're going to fill out the rest of that summary sheet. Make sure that you complete the bottom portion, the matching, where you're going to match the cell cycle checkpoint to an issue that may have occurred in the cell cycle. And when you're finished with the cell cycle